Coagulase test. In the laboratory, the coagulase test is used to identify Staphylococcus aureus and differentiate it from most of the other species of Staphylococci. The coagulase is believed to function in vivo by producing a fibrin barrier at the site of Staphylococcal infection, so it will localize the pus in form of abscess. That's why carbuncles, furuncles, and other localized pyogenic infections are common in Staphylococcus aureus infection. What is the principle of this test? Coagulase is a protein of unknown chemical composition. It has a prothrombin-like activity. It converts fibrinogen to fibrin so that a visible clot will result. Coagulase is present in two forms. One is bound and another is free. Each is having different properties that require use of separate testing procedures. The bound coagulase is detected by a slide test. It is also known as a clumping factor. This is bound to bacterial cell wall. That means this bound coagulase or clumping factor is present to the cell wall of Staphylococcus aureus. That's why it is not free. That's why it cannot appear in culture filtrates. So whenever it comes in contact with plasma, the fibrin strands are formed between the bacterial cells and it causes the cells to clump into visible aggregates. The free coagulase test is detected by a tube test. Free coagulase is a thrombin-like substance and it is present in culture filtrates because it is free. But it requires to react with a serum substance known as coagulase reacting factor and form a complex, and then this complex will convert fibrinogen into fibrin and produce a clot. So what we need as a reagent for this test? We need rabbit's plasma with EDTA. It is available commercially in lyophilized form. We have to reconstitute it, and reconstituted plasma can be kept refrigerated. We always have to perform quality control while doing biochemical test. First, we have to check the quality of plasma, and that will be checked by the coagulability of plasma by adding 1 drop of 5% calcium chloride to 0.5 ml of reconstituted plasma. A clot should form within 15 seconds. Then we also have to check whether this rabbit plasma will give us positive result and negative result properly, so that we put a positive known strain, that is of Staphylococcus aureus, and negative known strain, that is of Staphylococcus epidermidis, respectively. How to do the test? First we will see how to do a slide test, then we will see how to do a tube test. The slide test: first, place 2 drops of sterile water or saline in 2 circles drawn in a glass slide with a wax pencil. Gently emulsify colony material from the organism to be identified in liquid in each of the circles. Place a drop of coagulase plasma in the suspension in one of the circles. Mix with a wooden applicator strip. Place another drop of water in another circle as a control. Rock the slide back and forth. Observe for the visible agglutination. A positive reaction will be detected within 10 to 15 seconds in form of precipitate and agglutination of the organism in the suspension. If test does not appear positive, you cannot declare it unless you have observed it for at least 2 minutes. The saline control should remain smooth and milky. Procedure for the tube test. Emulsify a small amount of colony growth of the organism in a tube containing 0.5 ml of plasma. Incubate at 35 degrees Celsius for 4 hours into the incubator and observe for the clot formation by gently tilting the tube. If no clot is observed at the time, re-incubate at the room temperature and again after 18 hours. A tube should be gently tinted and not agitated because this may disrupt or partially form clotted material. The tube tests that are negative after 4 hours should be incubated at room temperature overnight and read after 18 hours. Sometimes fibrinolysin produced by organism may also dissolve the clot soon after the formation. That's why we read the test for 2 times, one at 4 hours and second at 18 hours. This is a diagram showing coagulase light test or clumping factor detection test. You can see the coagulase positive organism on the right side and coagulase negative organism on the left side. If clumping of cells are there, the plasma will be coagulated and it is a positive test. If no clumping is seen, plasma has not been coagulated and it is a negative test. The upper slide, it is a positive coagulase tube test. That you can see a coagulated plasma or clotted plasma which is not moving upward on treating the tube, while the below tube is showing us that fluid is moving or plasma is moving upwards. That means it has not been clotted, so it is a negative tube test. Thank you.